Thanks for clicking on my talk. Okay, so we know that within streams, heterogeneous habitats can facilitate the co-occurrence of processes we typically thought of as competing. So this is a very idealized stream reach where we have different habitat patches, leaf litter, macrophytes, boulders, and some sediment. And those different habitat patches have different characteristics, right? So the leaf litter might have more organic carbon, macrophytes, they're producing organic carbon and oxygen. The boulders, typically they're in a riffly area, there's lots of gas exchange, they would have more oxygen. And the sediment, the structure of the sediment may develop low oxygen minimum zones. So we know that those different characteristics would foster different microbial processes. This is an idealized stream reach. An actual stream reach might look more like this, where you have an uneven distribution of those different habitat types. So here we have way more boulders than anything else. So the reach average rates of those microbial processes would be based on both what kinds of patches are there, but also the abundance of each patch, right? Now we can also think about how within a single substrate, the processes may be interacting with one another. So here we have the microbial process of carbon transformation, so GPP or gross primary production, and ER or ecosystem respiration, and then nitrogen transformation. So we have nitrogen fixation and denitrification. Now in streams, generally we think of our nitrogen fixers as photosynthetic cyanobacteria. And those photosynthetic cyanobacteria, they're performing primary production to generate ATP, which then is used to fuel their nitrogen fixation. So the factors that might increase GPP would also increase N fixation. So light and phosphorus would increase both processes. Here in this diagram, I should orient you that the dotted line means produces, the full line means consumes, the blue line means something promotes a process, and the red line or pink line means that something inhibits a process. Now, if we look at denitrification and ecosystem respiration, we know that ecosystem respiration will consume oxygen. We also know that the presence of oxygen will inhibit the process of denitrification. So if we have more ecosystem respiration consuming more oxygen, that might equate in the promotion of denitrification. So we know that carbon and nitrogen cycling can be determined by those habitat patch characteristics. And it's likely that also within those patches, feedbacks between carbon and nitrogen cycling processes are happening. So we ask, at the reach scale, does the abundance and diversity of stream habitats affect rates of carbon and nitrogen cycling? So we had an opportunity to investigate this question due to an ongoing project in our lab. So the Marcarelli lab from the years 2017 to 2019 went out and measured nitrogen fixation and denitrification in streams across the US, in 30 streams across the US. And of those 30 streams, 12 of them were NEON sites. So we were able to partner the data from those 12 sites with other NEON collected data to investigate this question. So what we did was we pulled data used to model metabolism. So we pulled that, and then we also pulled some SF6 reiteration measurements collected by NEON. We pulled a lot of nutrient grab sampling measurements, and then we also pulled aquatic plant survey data. So this is aquatic plant surveys, but it also characterizes what's actually in the stream bed. So the first thing we had to do was we had to use that aquatic plant data to calculate the proportional cover of each habitat within the stream bed. So this is a diagram of how NEON samples for this aquatic plant data product. What they do is they establish a transect across the reach, and then at each point along the transect, they categorize what aquatic plant is there. And if there's no aquatic plant present, what they do is they note the dominant substrate of that point. So using that data, we're able to then calculate the dominant substrate within each stream reach here. So that's the proportional cover calculation. And then what we also did was we calculated these indices of heterogeneity. So we calculated a Simpson's index using the proportional cover, and then we also calculated a Shannon's evenness index indice using the proportional cover. So now that we have a proportional cover, we're able to then scale the chamber rates we made on each substrate to a whole reach measurement of nitrogen fixation and denitrification. So these are the plots of those rates in these 12 NEON streams. So denitrification rate on the left-hand plot, and then nitrogen fixation rate on the right-hand plot, and then on the y-axis we have each stream in this study. So the denitrification rates range from 0 to around 10 milligrams of N meter squared per hour, and the nitrogen fixation rates were about an order of magnitude lower than them. Interestingly, the site that had the highest denitrification rates was the Rickery River, which also had the highest nitrogen fixation rates. So to compare rates of nitrogen cycling to carbon cycling, what we had to do was model metabolism in each of our streams. So what we did was we pulled those SF6 measurements collected by NEON, and then using the NEON Science reaeration rate package, which will be talked about by Kaylin later on in this session, which is awesome. Using that package, we were then able to calculate gas exchange. So here are some plots of the measured discharge in each stream versus the measured gas exchange rate. So the left-hand plot, it shows a very beautiful linear relationship between the discharge measurement and the gas exchange. So using that linear equation from this relationship, we're then able to extrapolate gas exchange at each discharge measurement for the full time series. At sites where there was not a very nice, beautiful linear relationship, so that's the site on the right-hand side of this plot, which was only 2 sites out of the 12. We just used a mean of the measured gas exchange numbers for all of the discharge measurements in the time series. The other thing we had to do was fill any missing data points. So what we did was we used an ARIMA model, which is a time series model, so that the results of that here are in the dissolved oxygen plot. So you'll see the very straight lines. That's the ARIMA filling the gaps in between the two points there. It's very much less obvious when there's a small time gap that needs to be filled. There's also an upstream sensor station and a downstream sensor station at NEON sites. The goal is eventually to do 2-station modeling. I'm working on that right now, but I forked the NEON stream metabolizer code, excuse me, but there's an error in the differential equation I've written. So what I'm showing is the single station models for now. So I've run those using the downstream data using a maximum likelihood estimator model with a fixed K input based on those relationships with the gas exchange. So the results of the metabolism modeling, what they show. So on the x-axis, we have date, which generally goes January through the January of the next year. So it's full year, with the exception of Caribou Creek, which is late May through late September because Caribou Creek freezes in the winter. And what we have on the y-axis is primary production in blue. So the circles are the actual model results, and the lines are just a lowest smoother. So that's just a linear regression that takes the nearest 10% of the data points to give you a smooth, continuous estimate of metabolism at all of these sites. And then the respiration rates are in pink. So the dotted line you'll see on the plots, that's the date we took those nitrogen fixation and denitrification measurements. So at some of these sites, we're taking those N cycling measurements during a time period where production and respiration are relatively low in comparison to production and respiration over the rest of the year. So that's Kings Creek, relatively low. Some sites we're at a peak of production and respiration. In Pringle Creek is an example of that. And some sites are relatively representative of what's happening throughout the year. So Rio Guiarte is an example of that. So that's just to say that these nitrogen transformation rates may or may not be representative of what's happening throughout the year at these sites, right? They're just a grab sample measurement. So looking at how carbon and nitrogen cycling correspond to habitat diversity, what we have here is on the x-axis, we have our Simpson's Information Index, which as that number increases, that represents greater habitat diversity. And then on the y-axis, we have either denitrification rate, nitrogen fixation rate, gross primary production, or ecosystem respiration. So in general, we see that habitat diversity on its own is not a very good linear predictor of most carbon and nitrogen transformations, with the exception of gross primary production, which as we increase in our diversity, we see a corresponding increase in gross primary production. So in general, we find that these relationships aren't linear. They're probably caused by a combination of factors and not just one predictor alone. So that's why we decided to take this predictive modeling approach. So the goal of predictive modeling as a data science method, the goal is to find a model that may incorporate multiple factors to describe your data best, without overfitting your model. So how we do that is we take all of the data we have, which is the nutrient grab samples, the substrate cover of each substrate in the stream, the heterogeneity metrics that we calculated, watershed area, and then also our GPP, ER, N fixation, and denitrification. And what we're doing is we're trying to predict which of those predictors are the most important to controlling either the response of GPP, ER, N fixation, or denitrification. So there's 4 predictive models that we're creating. We take all of that data and then we slap it into either a training set or a testing set. 80% of the data goes into training, 20% into the testing set. And then of the training set, the testing set gets reserved. So that's our overfitting safety net, is that we're not touching the testing set, but we build the model on the training set. So within the training set, we're using a bootstrapped resampling method to jumble up our tiny data set into different new data sets that we then train the model on and get a comprehensive look at which model is best. What we're doing is we're training an array of models, linear, nonlinear, and then also tree and rule-based models to find the model that predicts the response best. So what were the most important predictors of carbon and nitrogen transformations? For denitrification, we found that the most important predictors were dissolved organic carbon, ammonium, and then also total dissolved nitrogen. And those are all factors that we know are very important to controlling denitrification in streams. So that's good that our predictive model agrees with what we already know are very important factors for denitrification in streams. On the contrary, for nitrogen fixation, what we found was that watershed area, the amount of pebbles in the stream, the amount of silt in the stream, the proportional amount of silt in the stream, and then also the proportional amount of the dry riverbed, those were the most important predictors to controlling nitrogen fixation rates. So that's interesting. So other than watershed area, we haven't yet included any other landscape scale characteristics into this model, but we're going to explore that further. For GPP and ER, what we found is that the model results predicted that many predictors were important to determining those rates. I think that's because our data set for these metabolism metrics doesn't have a whole lot of variation between the results of GPP and ER, so many of the predictors are determined as equally important. That will change as I am adding metabolism results to this data set. So that is inconclusive so far. In terms of next steps for this data set, I want to complete the metabolism modeling data set using 2-station metabolism models. And then also what we have is we have rough stream substrate maps that we made at all 30 sites as part of the study. So we'll be able to get a very rough substrate characterization of all of the sites. And then we're going to compare the sites that are neon sites, the maps we collected, to the real neon data to see how accurate it is. If it's okay, what we're going to do is then use the data we have from our rough maps to scale up our chamber measurements to stream reach measurements at all of the 30 sites in the study. So then we'll have much more data points, which will be great. Also, I'm interested in exploring more landscape characteristics as possible predictors using some GIS data sets that we just assembled, so that should be good. Preliminary takeaways from the study. 1, habitat type appears to be a better predictor of reach scale rates than habitat heterogeneity. 2, landscape characteristics might also be really key here. So that's where we're headed. In terms of neon data and the value here, the giant breadth of neon data, the breadth of data that neon collects, allowed us to explore many, many predictors, way more than we would have been able to feasibly measure ourselves. And then that allows us to narrow down what we focus on at those other study sites where we don't necessarily have the same breadth of data that neon collects. So super valuable. This analysis likely would not have been possible without the data that neon collects. So in conclusion, there are many undergrads, a small army of undergraduates who worked on this project, and I would like to thank you all. The funding for this study, the nationwide study as part of the Mark Rowley Lab, was provided by the National Science Foundation. NEON, who gets also funding from the National Science Foundation, provided a lot of the data used in this project. The code I used to both pull neon data and model neon data, model metabolism neon data, I'm wrapping into an R package that I'm very unimaginatively calling Neon Metabolizer. You can get it on my GitHub page right now. It's very beta, but the goal of that is to then eventually publish a finished our package that will allow people to model two-station neon metabolism easily by wrapping a whole bunch of other functions into the same house of functions. Drop me a line, catch me at JASM if you want to say hi in person. I'd love to talk to you about any of the stuff here or other stuff. Okay, thanks. Bye.